This is episode 259 of the Stem Cell Podcast, Regenerative Neurobiology, with Dr. Malin Parmar. Hello, everybody. We are Daylon and Arun. Welcome back to the Stem Cell Podcast, where we culture knowledge and stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Stem Cell Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Malin Parmar from Lund University. She's on the podcast to talk about her research on neuronal differentiation and Parkinson's disease. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, let's chat a little bit about ISSCR 2024 coming up in Germany in just a few months. It's, of course, going to be the most clinically focused annual meeting of the ISSCR in 20 plus years. Fueled by basic research and unparalleled development of new technologies, we are indeed seeing more stem cell-based clinical trials than at any point in history. The ISSCR annual meeting is an incubator, a catalyst for new advances at each point in the research continuum. Join innovators at this stem cell event and forge meaningful connections with our global community of academic and industry leaders. And indeed, we are very excited to be attending ISSCR 2024 in Germany in just a few months. And this is an episode that's going to be focusing on one of the preeminent scientists in the ISSCR. That's, of course, Dr. Malin Parmar, who has been involved with the society for some time now. Regenerative Neurobiology is the title of the show, and we have some papers here on the Roundup that are very relevant to Dr. Parmar's line of work. We're actually going to start off with a cell paper titled Human Fetal Brain Self-Organizes into Long-Term Expanding Organoids. This is something that's actually gotten quite a bit of attention recently. Notable authors of this paper include the one and only Hans Clevers, organoid aficionado and extraordinaire himself. And last author is Benedetta Artogiani, both, of course, from the over there in the Utrecht in the Netherlands, Prinses Maxima Center for Pediatric Oncology. As we know, Dr. Clevers has also moved on to Roche as well. So let's chat a little bit about what this paper is all about. It's gotten a lot of press, and rightfully so. This is establishing fetal organoids, fetal brain organoids, sort of in parallel fashion to what's already been done by many groups from human embryonic stem cells and human-induced pluripotent stem cells. And some of the really striking things about this particular paper are how they've been able to characterize and carry forward these organoids for many, many months and keep them growing long term. Certainly, there are a number of ethical considerations to think about when it comes to a study like this, utilizing human fetal brain tissue and deriving organoids from them. But let's dive in. So we know that human brain development is this really intricate process. It's got this massive neuroprogenitor expansion where you're actually forming the multicellular tissue architecture, right? And what you can do through not just brain organoids, but all sorts of fetal tissue organoids and just organoids in general is just take a small sample of the somatic tissue and then get those clumps of cells to grow very long term. I mean, this is what Dr. Clevers had demonstrated a long time ago, initially in the gut, has been carried out in a number of different types of tissues. And here they're extending that same approach to the fetal brain. So as of now, until this paper really, brain organoids could only really be truly established from pluripotent stem cells, like I mentioned. And here they're actually showing that healthy human fetal brain in vitro can self-organize into organoids, or what they call phebos here, which is phenocopying different aspects of in vivo cellular heterogeneity and complex organization. So the cell types and the connections that you would expect to find in the fetal brain are recapitulated in the fetal organoid, or the phebo, in the fetal brain organoid itself. Like I said, the fetal brain organoids can be expanded over long time periods, many, many months, many passages, and really an exponential expansion in terms of the biomatter that you can actually get from the initial brain organoid. These things are able to really grow out quite impressively. The growth of these phebos actually requires maintenance of tissue integrity, which ultimately is ensuring the production of a extracellular matrix niche, ultimately endowing this massive expansion of these phebos. And the other important thing was the PHEBOs, the fetal brain organoids that they derived here, were somewhat heterogeneous, meaning that they weren't all from the same type of brain tissue from the fetal tissue. So you have different areas of the central nervous system that the organoids were derived from, including the dorsal and the ventral forebrain, as well as I believe some spinal cord tissue as well. So you can preserve their regional identity. These separate organoids from the separate regions of the central nervous system are able to maintain their identity independently. And you can use this to probe aspects of positional identity in the central nervous system. And then the next part of this is the application. And I think this is part of the reason that it ultimately was published in Cell is, of course, not just the exciting development of these things, these fetal brain organoids, but also their ultimate application for the generation of syngeneic mutant Phoebo lines for studying different types of brain cancer and pediatric neural disorders. They did this through CRISPR-Cas9, which was very easily intersected with these fetal brain organoids, that was really cool to see. And ultimately, it's a really unique model system to study central nervous development, brain development in a way that complements the existing systems that are already out there in human embryonic stem cell derived brain organoids that we talk about all the time here on the show. I'm actually very surprised that something like this hasn't been done already. Maybe it has something to do with the ethical considerations, the regulatory hurdles that they had to tackle to actually make something like this happen. But I'm glad we're here. I'm glad we finally have this model system to help better interrogate the development of the human brain.
Yeah, this is a cool study. Got a ton of press, I think, probably because the fetal origin of the tissue always spices things up. And yeah, I think that's a critical advance here, is that showing, as you said, maybe for the first time, I don't know, for the first, I think, notable time that made such a big splash, they're able to get these long-term sustaining organoids. And I don't know the answer to your question, but along the lines of, why hasn't this been done before? But I would guess that a lot of the lessons we learned from the in vitro culture of pluripotent stem cell derived neuronal cell types, namely the recipes that sustain them and allow them to proliferate and you know become all the different derivatives specialized. I think that a lot of those lessons were applied here. uh, I don't know. I think this is a great study. I'm kind of left in a place like, okay, where do we do with this? I like the idea of having like I guess you would call it an adult stem cell or fetal stem cell or whatever, not a pluripotent stem cell derived culture system that can, be sustained over the long term. So pretty much one off, you get some fetal brain tissue and you can set up these cultures and systems and platforms. So I think that it's robust for experimentation. But I think the most exciting thing right now in vitro culture of neuronal cell types is still pluripotent cells. I don't think anyone would really disagree with me there. Uh, I would be really interested to see how these fetal brain organoids are used as a kind of a benchmark for in vitro culture. With the bona fide cells, you could explore things like whether or not that maturation paradigm is the same. The great challenge in neuronal differentiation from pluripotent stem cells is, of course, as we talked about just in the last episode, is getting these to advance to later maturation stages where either phenotypes or disease will manifest. So I wonder if these fetal brain organoids can mature on the same time scale as a in vivo. So that'd be a really important question, I think, that you could use to answer these. I think, though, still a bit limited in terms of what type of organoids you can get from the fetal brain. Here, we're talking about forebrain, where we talk about assembloids in the pluripotent stem cell system, where we're getting multiple cell types. So I know you alluded to the spinal cord tissue there, but I think the majority of the cell types they're working here with were forebrain. So I think this is a big splash. I think there is a lot that could be done on this platform, but I'm still, I would say, a lot more excited about pluripotent stem cell derived systems.
Yeah, I think we're a little biased towards the pluripotent stem cell applications. I agree with you. I mean, on the contrast that there are certain folks who just simply do not believe in the applications of pluripotent stem cells. They think they're a completely artificial cell type, and having something like this, which is truly primary tissue derived organoids, I think those folks might think this is the only way to go. I completely agree with you. I think the real power of this, in my mind, maybe for both of us, is that this could be a benchmark, a really important benchmark to compare your pluripotent stem cell derived neuronal organoid cultures, because this is the real deal, right? This is derived from true human fetal tissue. And I do think there are some other things that they want to do with this that they actually discuss in the limitations of the paper. I think one thing that would be really cool, they alluded to, would be deriving these fetal brain organoids from diseased tissues, early fetal disease tissues, to see how that impacts their developmental trajectory. So I think there's a lot of interesting applications for this kind of work. But I think the other part of this work, in contrast to the pluripotent stem cell culture, is that this may be a little bit more heavily regulated, and ethically considered, right? Because anybody can really do iPS culture now, right? You don't even have to use human embryonic stem cells to derive brain organoids from, and iPSCs are much more limited in terms of their ethical considerations. Folks around the world can use them, but stuff like this, when it comes to primary fetal brain tissue, I think there's a lot more restrictions when it comes to using this kind of stuff.
Agree. Yeah, I mean, access. That's going to be the thing. This is very niche. It's going to be accessible to few, although the expandability, self-renewal, I guess, of these cells means they can be distributed. So definitely a lot there, but pretty niche, I would say. Highly regulated, as you said. The opposite, although we're staying in neural, I got a story that's kind of on the other side of the pole here. Instead of fetal, we're talking about adult disease. And instead of niche, we're talking about something that I think we're all very familiar with, and that's COVID, right? And specifically the neurological manifestations of COVID that we're all aware of. I think very commonly, a lot of people could tell you about how they had the experience of the anosmia or dysgeusia, however you say that word. That's the loss of smell and alteration of taste. I didn't have that one, but I mean, I'm glad, because that would have really freaked me out. A lot of people I know struggle with that, but also more, I guess, severe, you could say, manifestation, seizure, stroke. There's a lot of Guillain-Barré syndrome. And more recently, it's emerged that patients who had struggled with long COVID are also at increased risk for neurological and psychiatric disorders. So it's a really big deal. I think, we were so focused on the acute phase there, death effectively. And now that we're out of the woods there, I would hope we have to wrestle with all these long-term phenotype, the manifestations and increased risk. And of course, based on those phenotypes and the presentation, it was believed that there was this tropism, neuronal tropism. And Madeline Lancaster and another paper in the same issue, Cell Stem Cell, back in 2020 from UPenn, Scripps, and Sanford Burnham, also showed, like Madeline Lancaster's group, that choroid plexus and the central nervous system were permissive and really susceptible, highly susceptible to infection with SARS-CoV-2. So that was like, I don't know about problem solved. It was like, okay, there we go. There's the entry point into this neural sphere, neural milieu, and may account for some of those neuronal manifestation or neurological. But it's still controversial as to what cell type specifically CoV-2 targets. All right, so this is a story from the lead here, Shubing Chen, who just continues to pump it out. But in collaboration with some other big hitters, Rob Schwartz, who's at Weill Cornell, so David Ho, who's up at Columbia now, and Lorenz Studer, of course, who works closely with Shubing on a lot of things. Specifically here, they were looking at, makes sense, dopaminergic neurons. They use this platform to explore the tropism, SARS-CoV-2, and show that indeed midbrain dopamine neurons, and importantly, not cortical neurons. So it wasn't this like pan-neuronal tropism. It's specifically midbrain dopamine, also choroid plexus, of course, which is known, but not cortical neurons are susceptible, permissive to SARS-CoV-2 infection. And that infection triggers this inflammatory and cellular senescence response. The group then went on, as Shubing is wont to do this high throughput screening, identified a bunch of already approved drugs that can rescue the cellular senescence. They mitigate or preclude the infection in the first place. And they also here, and this is the key to lend some kind of clinical relevance, they identified this signature of inflammation, cellular senescence, the same thing they saw in culture, as well as low levels of SARS-CoV-2 transcripts in the actual substantia nigra of CoV-2 patients. That's the store of these dopaminergic neurons. So evidence that SARS-CoV-2 was actually in the patient brains in the region where these dopaminergic neurons are and cause inflammation. Also found reduced dopaminergic neurons in patients who had severe COVID. So these are patients, all of whom presumably died from COVID, and they saw the disease was present in the brain. So this is like a pretty straightforward, but I think really elegant study with, I think, tremendous implications and impact, suggesting that it's this specific cellular component, right? These dopaminergic neurons that are also affected in Parkinson's disease, right? Which is really scary because you wonder now if there's this occult or latent PD potential in like millions of people that didn't have it before. Anyone who may have suffered from one of these neurological manifestations of COVID-2 may be at risk for Parkinson's, Arun? I mean, I don't know. I wouldn't say that that's the direct takeaway from this study, but certainly among the implications.
That is a potential connection that you can definitely make here and has been floated around on the Twitterverse or Xverse, whatever you want to call it. Yeah, I mean, anytime you're talking about dopaminergic neurons, that is the ultimate connection that you're making with Parkinson's, right? So we'll see. I mean, hopefully that doesn't come to fruition many years down the road, but we'll have to definitely keep an eye on those individuals who have been affected by the disease. A few things to consider here. Obviously, one, just reflecting on the productivity of Shubing, your neighbor there. She is just unbelievable. This is Shubing's world. We're just living in it. I feel like we're covering one of her papers every single roundup here on the show. So congratulations again, Dr. Chen. A few other things to think about. And I actually was reflecting on the parallels between this sort of work and the cardiac work that was actually coming out during the time of COVID as well. Because, iPS-derived cardiomyocytes were also used to model cardiac-specific SARS-CoV-2 infection. And one of the limitations here in this paper that was also reflected on the cardiac studies was that the detection of the SARS-CoV-2 viral antigen in the primary tissues is still controversial, even to this day. And in fact, they mention it here, they're still trying to generate electron microscopy to actually assess evidence for the direct viral infection in the substantia nigra neurons. This is the same deal with the cardiac. I mean, there's just not a whole lot of direct evidence through electron microscopy, for example, of SARS-CoV-2 actually entering cardiomyocytes and cardiac tissues. So this is still an ongoing area of study to figure out what's the exact cellular target and cellular mechanisms of infection in both of these primary tissues, both brain and cardiac. One other thing I thought that was also very interesting here, and this is also a reflection on this concept of drug repurposing that we talked about on the previous show. One of the drugs that they identified in their screen to actually alleviate SARS-CoV-2 infection and the downstream neuronal manifestations was imatinib. Imatinib, this amazingly popular hallmark tyrosine kinase inhibitor that's been around for many years now, which is one of those hallmark treatments for CML. That happened to be the drug that actually alleviated the SARS-CoV-2 induced senescence of the dopaminergic neurons. So, hey, you never know what you might come up with in these drug repurposing studies. And I'm assuming that's some kind of off-target effect that imatinib is happening, because I can't think of the exact mechanistic reason as to why that would happen. But hey, I mean, these are small molecules. They have all sorts of off-target effects, right?
Yeah, so I mean, I guess it's a win if you had CML while you caught COVID and were being treated with imatinib, you might be spared the Parkinson's, or for that matter, metformin, right? All those pre-diabetics. But I think, yeah, that was the bummer for me about this is, of course, I mean, what do we expect, right? You can't rescue the effects of the infection after the infection has been cleared. But that to me was why I had such a pessimistic view after this. Is like, okay, now that we're through the actual acute phase of the pandemic, now we have to live with the mess it left behind. And there's really, I don't know that there's a way to prevent, um, you know, those people who may already have been affected by this neurotropism, but certainly moving forward in the future, it's something that we can watch out for. I don't know. For me, also, the big takeaway was that I was so comfortable in the idea that this was predominantly a vascular disease, based on even with the neurological manifestations of stroke, you could attribute that to cardiovascular and also even the chord plexus. I don't know. For me, it was like this idea of the cerebrospinal fluid. I don't know. It's something that conveys fluid similar to vessels, but I think the jury is back here now with the idea that there's very deliberate, I think, neuronal tropism and neurological manifestations that cannot be denied here. And I guess that's the important thing here. And to your point about whether or not the virus directly is shown to affect in the body, they made a glancing blow at it here, perhaps not definitive, but I think that's what's the value of these in vitro studies. The whole point where we started here with the platform is to show, as many papers showed in the first days following COVID, that this cell type, that cell type, the other is permissive to COVID infection. If it's permissive in vivo, I'm going to assume that it could also happen. I mean, in vitro, I'm assuming it can also happen in vivo. So I'm gonna assume that we don't need to wait for the electron microscopy, that this is real, and it's something that we need to keep our eye on with all these long COVID patients. I don't know how you work them up, but definitely something that we should be aware of.
Yeah, definitely. I think this is something that's not going to go away. And we're going to have these long COVID clinics popping up across the world, across every single hospital. I know we have them here at my institution. And I think this is just going to be a growing concern as these individuals get older, and, just keeping an eye on their neurological manifestations of this disease for long COVID. And who knows, maybe this is even something that, I don't know, Blue Rock might want to get into as well. Hint, hint. Um, Moving on to another stem cell paper. This is very different from, actually maybe not even that different from the studies that we were talking about, because this is another model system, another organoid model system that was actually also used as a SARS-CoV-2 model. The title of this paper is Human Conjunctiva Organoids to Study Ocular Surface Homeostasis and Disease. This is another Hans Clevers paper. Congrats again, Dr. Clevers. And the first author here is someone who we've actually talked about on the show previously, from the Clevers lab, Marie Bagné-Halouet, who has established a number of different organoid models and that we've discussed here on the show. So again, another organoid system, another primary derived organoid system. This is not stem cell or iPS, pluripotent stem cell derived organoids. These are primary tissues from the conjunctiva, from the inside of the eye, to actually make these long-lasting organoids, both from mouse and from human. So let's dive right into it. So we know that there's this conjunctival epithelium that's actually covering the eye, and it has two different cell types, these mucus-producing goblet cells and the water-secreting keratinocytes, which actually present mucins on their apical surface, and I think are involved in moistening the eye and that sort of thing. So here they're actually describing a long-term expansion, similar to the first story, long-term expanding organoids and an air-liquid interface that's actually representing both mouse and human conjunctiva. Then they actually characterized the organoids that they made here using a bunch of single-cell expression analysis of comparing these tissues to other primary and cultured human conjunctiva, and showing that the keratinocyte populations actually express a bunch of different antimicrobial peptides, and identifies these conjunctival tough cells, a subpopulation found in these organoids. And this was their foundation to the second half of the study, where if you have some antimicrobial properties, maybe you can utilize those in the context of addressing disease and infection, that sort of thing, right? So they found that this IL-4, IL-13 exposure increased the goblet and tough cells differentiation and actually modified the conjunctiva secretome. And then moving on beyond that, their human NGFR positive basal cells were actually identified as these bipotent conjunctiva stem cells. So they can turn into multiple cell types from these stem cell populations. And here's, I think, where things got really interesting and probably why it elevated to the level of cell stem cell is ultimately they utilize some of these properties and some of their observations to treat these conjunctival cultures and organoids with different types of pathogens, different types of viral pathogens, including the herpes simplex virus 1, HSV1, human adenovirus, and relevant to what we were just talking about, SARS-CoV-2, which is, this is thought to be one of the avenues of infection for SARS-CoV-2 is actually via the eye and the ultimately, traveling down to the nasal cavity and all this kind of stuff. So perhaps very relevant model system to actually study SARS-CoV-2 tropism. We were just talking about that, right? So, and then, the usual stuff, you treat the organoid system with the viral agent, and then you try to rescue the infection, right? Just what we showed in the previous paper from Schubing and colleagues. The herpes simplex 1 virus infection was reversed by a cyclovir addition, which is, makes sense. This is an antiviral. Whereas the adenoviral infection, which actually lacks an appropriate drug therapy, was inhibited by another type of antiviral compound called cidofovir. And then they looked at the transcriptional programs that were actually induced by the human herpes simplex virus 1 and the adenovirus, compared their transcriptional responses to the different viral pathogens. And then the very last thing that they did here was something that I think was a little bit far out there. Still trying to wrap my brain around exactly why they were doing this. They transplanted these conjunctival organoids. So they generated the human conjunctival organoids and then transplanted them into the mouse, ultimately showing that these human conjunctival organoid cultures can enable the study of pathophysiology. All right, that's both what they showed in vitro and also to some extent in vivo in the transplantation culture. So really just a broad study, very broad study, just ranging from the initial derivation of the organoid itself to the characterization transcriptionally, and then ultimately the application, which is the infection of the virus, and then treating it with the antiviral compounds to actually rescue the infection. So a really, really broad range study. And I'm sure this was a very long, multi-year piece of work, but absolutely reflected in the caliber of the study that it's in.
Yeah, I mean, why did they put the human in the mouse, Arun? Because they could. Because they could.
Yeah,
All right, and that's what
sure.
Hans does. He does what he can, and he doesn't stop. This story, as you said, years in the making, and a follow-up actually. First author on this, Marie Banier. Hello, sorry about the pronunciation. All the vowels are like right next to each other in that name, so I'm not doing a good job, but I'm an American. Anyway, Marie Banier was also first author on that paper just about 3 years ago now. That was the lacrimal organoids, where they generated the organoids, again, similar methods, and showed that they could generate tears. So this is a story from Marie there, with Hans Clevers that she's been working on for probably close to 5 years. And this conjunctiva one is like the feather in her cap for defending her thesis, Arun, if you can believe that. So these two stem cell papers that made a big splash about the eye, and she's moving on, co-corresponding on this, so presumably starting her own lab, I would guess, some kind of ophthalmology tie-in here, because she's really focused on the eye. And as you said, the range of this, going from derivation all the way to application, I think speaks to the maturation of this work. There's still some ways to go, of course. You talked about in vivo. In vivo, these components of the eye are actually closely associated with a lot of other cell types, namely, the endothelium, stromal cells. Also, the immune component, as you're alluding to there, is critical. So I don't think we're at the point of making, I don't know, whatever you make with these in vivo, making that in vitro completely, but I think that it's certainly evidence of the maturation of the work and creating, I think, a whole field out of these eye organoids and demonstrating that they have some really important applications.
Yeah, I think a lot of different applications here. The transplantation study is still a little confusing to me. Maybe we'll have to have Marie back on the show or on the show to chat more about that. If you're listening, Marie, we'd love to have you. I love talking about your work. But indeed, I think a lot of new avenues that are opened up as a result of these studies. Anytime that you're establishing a model system, you become the expert in the applications of that model system, right? And I think this is an amazing foundation for a new lab to come from Marie's work, right? She is the expert. She set this system up, and she, above anybody else, knows how to use it, right? Maybe Hans too, right? But a few, like you're saying, there's a few limitations here as well. You mentioned one of them. It's a reductionist model, right? It's not really encompassing all the blood vessels, nerves, stromal cells, immune cells that are actually present in the real deal, but are absent in these organoids. But I'm sure they'll be building those cultures into the model system pretty soon.
Yeah, I mean, I just realized, I think my new theory is Hans, he went to Roche because he just ran out of organoids to make. I don't think there's anything left. But I guess Marie is going to carry the torch, so to speak. We'll wait to see what she does. And yes, please get on this show, Marie. Open your lab with a splash. Get you to talk about it. Anyway, moving on to something that's not so niche as these lacrima or conjunctiva, the heart, right? This is the kind of target number one in pluripotent stem cells, but this is not exactly a pluripotent stem cell story. It's more of a developmental story, but I think it speaks to all that potential that we hoped for in the heart that has not been realized, tragically. Arun, get to work. Anyway, this is about epicardium and the difference, the delta between fish and mammals, effectively. Zebrafish, which is Arun's favorite model, which we've talked about a lot, has this capacity to regenerate the heart throughout adulthood, unlike the mammals, where it's been shown in mice that they actually can regenerate, but only in the early first week or so, neonatal stages. But in the zebrafish, that regeneration is mediated by the epicardium, which is this layer of cells that envelop the heart muscle and are critical for regeneration, but also really important during heart development. In the context of adult injury, heart injury, these epicardial cells are activated. They re-enter the cell cycle. They re-express a lot of these developmental genes. And they're among the first cell types that migrate to the injury. They spit out a bunch of extracellular matrix. And then they also act as like a signaling hub, coordinating the activity and signaling between other cell types in the region. And then they invade the actual subepicardial tissue, generating perivascular cells and fibroblasts. So they support this regenerative process both like as an orchestrator, as well as directly by secreting proteins and having cells that actually contribute to the architecture of that repaired heart. So across a lot of species, the whole idea of regeneration, I think, is steeped in this notion of the reactivation of a developmental program. And that's because a lot of the factors that you see expressed during regeneration are also expressed during development. And that's true of the epicardium as well. And also functionally speaking, that all those processes that I just described during regeneration in the fish, that's kind of what they do in development. They do very similar things. But this idea that regeneration is really just the reactivation of a developmental program remains controversial. And that's in part because in mouse or in mammals, using mouse as an example, the markers that you find during embryonic heart development versus injury activated regeneration or response in the epicardium are very different. So the question there is the mouse or are mammals different in regeneration or response versus development because they've lost this capacity to regenerate? And in the fish, it's the same. And that's important because If so, then it's really just a matter of restoring or recapitulating that developmental program, endowing the adult cells with the developmental program. So the question that was asked here by Tatiana Sauka Spengler and Paul Riley, who are at Oxford, is using the fish as an example. Are the adult and developmental programs the same in epicardium? And it's important because of what I just alluded to. So they compared the transcriptome, also the epigenome of the developing epicardia, as well as regenerating epicardia in the fish. And what they found is that they're different. I mean, bottom line, they're different. Looking at specifically these epicardial enhancer elements and using them to infer the genetic programs that drive development or regeneration of the heart, they show that these were really different. And then built on that to show that the specific program that's key to regeneration in the zebrafish, if they target that using CRISPR, here showing that if they target HIF1AB, NRF1, TBX2B, and ZBTB7A, like none of which I've heard about. Maybe HIF1AB, although that specific one I haven't heard about. So these are kind of new to me. They knock those out, and they show that the recovery, the regeneration is impaired, right? So the takeaway for me conceptually is huge. The science here is strong and solid. There's some work left to be done, I think, to really build out the link, mainly the question of whether or not loss of the other program, the developmental program, has a role. But the key here conceptually, I think, is really important, which is that my takeaway is that it's not, we can't just, enforce a developmental program in mammals necessarily and restore that regenerative phenotype that we see in zebrafish. That maybe it's a related but independent pathway that converges on some of the same cellular phenotypes and transcriptional circuits, but I think it's important to recognize that this is it's a whole new process.
Yeah, there's a lot of different elements here. I think it's beyond just the developmental programming, just at the genetic level. It's the epigenetic level that you really have to take into consideration here. And this is, I think, one of the major strengths of the study. I totally agree with you. I think it's more than just putting in a gene, putting in a particular enhancer or whatever from the zebrafish into the human and restoring cardiac regeneration in that way. I think you have to intertwine that with some of the epigenetic pathways as well. I really like the study, and as you might expect with any sort of cardiac regeneration zebrafish study, Ken Poss is cited about 15 times here, which, not surprising. Really cool work. I think one thing to think about is and reflect on is the epicardium, this layer of the pericardium kind of on the outside of the heart, which initially was kind of cast aside in favor of the more sexy myocardium, this muscular layer of the heart that does the dirty work, all the work of pumping the blood throughout the body. But now we know through years and decades of intricate basic science work, many of studies have been done in DevCell actually, that the epicardium has an incredibly important role in regeneration and restoration of proper cardiac function, especially early on during the developmental process. So, so shout out to you, epicardial folks. Much love.
Yeah, for sure. And I mean, I like to think of the failure of epicardium to restore the heart in mammals as, just something that was lost over the course of evolution and that became maladaptive, became more of a stopgap. And if we could perhaps maybe reactivate some of these genes and transcription factors that we see in the zebrafish in this independent adult regenerative circuit, maybe we could skew a few of those epicardial progenitors away from fibroblasts and maybe to more constructive fates, and actually solve this thing once and for all. Arun, again, I say, get to work, my friend. But for now, we're going to have to forget about the heart and focus on the mind with our guest, Malin Parmar, who's going to talk about her work with neural differentiation in just a minute. But before we get there, I have a quick message from STEMCELL Technologies, who invites you to find a pathway into the peripheral nervous system with STEM-diff neural crest differentiation kit. Whether you need Schwann cells, sensory neurons, or sympathetic neurons, STEM-diff neural crest differentiation kit enables high-purity generation of neural crest precursors from human pluripotent stem cells. Learn more at www.stemcell.com/NCKIT. All right, everybody. With us today, we have a special guest from Lund University, Professor Malin Parmar, who's also New York Stem Cell Foundation Robertson Investigator. Dr. Parmar's research aims to understand cell fate specification in the developing brain and in human neural progenitor cells using cell-based models of neuronal differentiation. Her lab's current focus is to learn how to direct and efficiently drive controlled differentiation of human stem cells into subtype-specific neurons. The ultimate aim there is to develop these cells and technologies for use in brain repair with a specific focus on Parkinson's disease. Dr. Parmar, thank you so much for joining us today on the show.
Yeah, great to be on.
Yeah, thanks for being here, Dr. Parmar. Let's start off with the big picture. You're, of course, working on bringing cell-based therapies for neurodegenerative diseases to the clinic. But I mean, in addition to the glamorous translational work that you're doing, you're also studying the basic mechanisms of brain development using pluripotent stem cells. So before we actually dive into the details of the clinical trial, could you actually give us a broad overview of the basic developmental neurobiology studies that your lab is focused on and just the overall scope of your lab?
Yeah, so I don't know how glamorous is translational science is, but for my experimental science, I'm a developmental biologist by training, and my PhD was on forebrain development. And I've always been fascinated how these processes are controlled, how cells are very similar, all of a sudden become different. And this is what we used a question that I worked on 20 years ago and that I still work on today. When it comes to stem cells, it becomes an interesting mix between developmental neurobiology and stem cell biology, because what we learn from the developing brain, we can test and see if we can use the same signals and mechanisms to control the differentiation of stem cells. So in a way, stem cell differentiation is also a type of developmental biology where we mimic the cues in the developing human brain into our stem cell cultures. So my lab do a lot of these kind of parallel, how do cells form and develop in the brain, and how does this happen outside the brain when we either take them from the brain and culture them outside the brain, or when we create them from stem cells or via direct reprogramming of skin cells, for example. So that's the one main focus. Other main focus is brain repair, and for that the cells need to go back into the brain eventually. So there's also a lot of basic experimental questions regarding what happens to an immature new cell when it hits a existing neural circuit. How does it integrate? What cells do they communicate with? What do the old cells in the brain think about the new cells being placed there? Do they recognize that these are new and young? Do they train them? Do they kick them out of their networks and things like that? So kind of the stem cell biology part is in the middle, spanning then the developmental aspects and the more neuroscience aspects.
Yeah, I mean, you're like a lot of the leaders that are guiding the translation of stem cell-based therapies in the present day, in that, as you said, I mean, your initial interest was a basic developmental biology, and current interest still is basic developmental biology of the brain. And I think also, like so many in that generation of scientific leaders that were coming into their own at that time, at the turn of the millennium, when pluripotent stem cells were first derived. They provided a means for unprecedented insight into human neurodevelopmental, in your case, neurodevelopment and disease, and that was irresistible. And here you are in the midst of driving transformative changes in the way we approach disease, which I know maybe your initial interest was the basic science, but it's all ultimately about the human condition and disease and repair. And we're going to come back around to that work specifically in just a minute. But first, just more generally speaking, as someone who has traveled that arc from the initial days of derivation, where the questions were all so basic, just how do we get this cell into that, and how do they work together? What do you think has been the most or one of the most pivotal steps in advancing the field to this point? And also, what is or what are the greatest challenges left to overcome?
So I'm actually older than what you the way you say, because my work didn't start with the big changes in the field of being able to grow embryonic stem cells. I was a PhD student when it became possible to take cells out of the brain and culture them, and we call them neurospheres normally at that time. So there was very, very basic questions regarding what happens if you take a cell from one area of the brain, take it out in the culture dish. Does it remember where you come from? Probably not, but it turned out that they did. But at the time, we thought that once you take them out of the brain, they have no positional information. And then if you take them from one region and put them back into another region of the brain, what happens to them? So it was a lot of these moving cells around and studying their phenotype in vitro and in vivo. And from that, we learned a lot about what happens to a brain cell outside the environment of the brain. And then it became possible to culture human embryonic stem cells. And that was, of course, a groundbreaking discovery in the field, especially with the human embryonic stem cells or the iPS cells, because all of a sudden we had unlimited access to the starting material to generate, in theory, any type of neuron in the brain from these stem cells. When that field developed, there's a lot of kind of technical aspects that made it possible. So when I was a postdoc at the Institute for Stem Cell Research in Edinburgh, they had just, Austin Smith was there and had just started to culture human embryonic stem cells. But every time we passed them, we just had fewer and fewer cells. It was really hard to get enough cells to do an experiment. So the ROCK inhibitor, for example, was a massive change, because all of a sudden, instead of doing this manual passaging where you cut the cells, you lost the cells, they died in each passage, you could generate a sufficient number of cells to actually do an experiment. And then in terms of the field of dopamine neurons, which is my main area of expertise today, we really tried a lot of different ways to direct stem cells into dopamine neurons, but our group, like other groups in the field, were unsuccessful until, again, developmental biology findings that showed that the origin of the floor plate cells, of the dopamine neurons, are the floor plate cells. And we knew that we needed to go from embryonic stem cell to floor plate to dopamine neuron, rather than from embryonic stem cell to neuroepithelial cell to dopamine neuron. So there's both kind of technical and biological advancements that propel this field forward, but one shouldn't underestimate the technical aspects and the ease of culturing cells today compared to what it was like 20 years ago.
Yeah, all glory to the ROCK inhibitor, the small molecule that it's made all of our lives so much easier over the years, right? So shifting, fast forwarding a little bit, diving a little bit more into the recent work that you've been doing. We actually recently covered your cell stem cell paper that you published, which actually discussed this preclinical quality, safety, and efficacy of the ESC-derived cells you're making for the treatment of Parkinson's, STEMPD, this trial that you're working on. I mean, this data is supporting one of a handful of trials in this area. I mean, there's stuff going around the world in this particular area using stem cell derived dopaminergic neurons for treating Parkinson's. We actually, maybe we had a show over at Blue Rock Therapeutics in New York a little while ago, which is, of course, also working on a parallel approach. Full disclosure, my boss is Clive Svensson here at Cedar Sinai, who I'm sure you know very well.
Yes, I
And
know,
he's,
Clive.
he's dabbling in the area too. So could you talk a little bit more about this preclinical study that you did in cell stem cell that was published recently, and how it's ultimately supporting the work of the clinical trial?
Yeah, so this is studies and experiments that we've done over a large number of years to convince ourselves and regulatory authorities that the cell product that we make is safe and efficient to use in humans. And I think it's very, very important, as this field is developing, that all the clinical trials that are initiated actually presents their preclinical data leading up to the approval of those trials. We all need to learn from each other's trials, and we need to give access to those preclinical data. That's absolutely necessary to propel the field forward. So to publish these data sets, both kind of serves as for education and scrutiny, but it also shows the massive effort that you need to undertake to go from an experimental finding, a protocol in the lab, to a clinical trial. So I think that they're great, and also the other Parkinson trials that are ongoing have also published similar papers leading up to their trial.
Yeah, I mean, talking about recent papers in our roundup, you've really been heavily featured. We just in the last episode covered an article that you played a prominent role in, this collaboration with Jürgen Noblich, where you guys modeled the human dopaminergic system with the midbrain striatum cortex assembloids. I mean, loved that paper. Not least of which because you had organoids dosed with cocaine. I mean, I was just picturing these organoids going on a cocaine bender. That was a little anthropomorphication there. A bit of an aside, but all joking aside, this is a watershed, in my opinion, because it not only enables the interrogation of the molecular pathophysiology of diseases like Parkinson's in a tidy model in vitro, but could also, as I alluded to, model the mechanisms underlying addiction and other perturbations in the reward pathway. Now, clearly, I think you were probably more involved with the Parkinson's end of that, although if not, please feel free to disclose. But regarding that Parkinson's end, at least, why don't you color in on how those assembloids can be leveraged in these kind of accessory experiments to advance treatments for Parkinson's disease?
Yeah, so I think organoids are very good model systems to study either aspects of development, because in Sweden where I work, we actually have access to human fetal tissue of certain ages that we can do some types of experiments on, but you can never really get the experimental detail and control or the extent over the extended time periods that you need to fully understand later development of the cells or the development of the functional maturity and things. We tend to do this in xenograft models where you put human cells into the rodent brain. And that is good because you really hook the cells up in neural networks. But it's not a very tractable system. And the organoids, where you can kind of mimic aspects or regions of the human brain, how they interact with each other, you can really get closer and closer to set up scenarios that will then inform you about how processes work in the brain. So in terms of cell therapy for Parkinson's disease, we can study the dopamine neurons themselves in ventral midbrain organoids, but for them to be therapeutically active, we need to know how do they connect with their target cells, how are they regulated, and those things you can study in assembloids or connectoids. Then it comes a lot of questions. The field is moving a lot into autologous therapies where we use patient-specific cells. And there it's a lot of questions to ask. What happens if you put a cell from a deceased patient into a diseased brain? And then you can kind of move cells from an organoid from a healthy brain and a diseased brain and do this mix and match experiments. And also here, the other cell types in the brain are important because in Parkinson's disease, for example, it's a spreading of the pathology. It involves other cell types like the glial cell in the brain. So you can never study this cells in isolation, and the organoid system is a very good model system to study both development, functional maturation, but also disease-related pathologies.
Yeah, it's a really powerful model system that's just taken the field by storm recently. We've had a few conversations with Sergey Pashka here on the show as well, who's certainly pioneered this area. But in addition to the pluripotent stem cell side of things, like you mentioned, your group is also exploring direct reprogramming and translational applications there. For example, you actually have this bioarchive paper that uses this 3D microculture engineering approach to enable intracerebral transplantation of mature neurons that are directly reprogrammed from patient fibroblasts. And I mean, there's certainly pros and cons in comparison to using the pluripotent stem cell approach if you're comparing direct reprogramming to that. But do you think direct reprogrammed neurons might become a parallel cell therapy down the road, parallel cell therapy to the stem cell based approach? So what do you think about that kind of alternative?
Yeah, I mean, it's hard to predict where the field will go, but I think that in the future there will be more options for cell therapy. So there won't be just one stem cell therapy for Parkinson's disease. There'll be many. And which one you opt for that is recommended for you depends on the type of disease you have, the stage of the disease, when the intervention is taken, etc. If you talk about, and patient-specific treatments have a lot of pros, of course. When it comes to iPS cells versus direct reprogramming, if we can learn how to handle it, I think is a better option. The problem is that we can't handle the conversion process properly. We can't make neurons are equally good. We worked for 10 years to get them to survive in the brain. So this bioarchive paper is, the crown of 8 years of failure and a year and a half of successful experiment. So there's a long way left, but if you can directly reprogram neurons that do not have a disease phenotype, I think they are a good alternative for autologous therapies. And with a direct conversion, those projects also lead to another area of the field that I work on and many other people have taken on, is in vivo conversion. Because if you can convert cells directly in the dish, you could potentially also do so in the brain. So in the future, maybe the best alternative is to just convert resident glial cells into neurons directly in the brain.
Yeah, I mean, I agree 100% with what you said there in terms of, I mean, it's obvious, I guess. There's going to be a lot of alternative therapies, a lot of approaches that are kind of in the same vein, but they're going to have nuance and there's going to be some differences and options. And in terms of like who has custody or ownership of those options, I just want to circle around a bit. And I mean, in terms of like funding is really what it comes down to. You and I both share a relationship with NICEF, the New York Stem Cell Foundation. They funded my postdoc, and you were awarded this coveted Robertson Investigator grant, which is meant to enable high risk, high reward research. You were awarded that in 2016, and I would be willing to venture that it paid off. High risk, but the reward has been significant and very fruitful. Particularly in the shape, I'm assuming in part, the STEMPED clinical trial, which was many years in the making, a lot of different funding apparatus, taking many forms ranging from the private philanthropic funds from NICEF and institutes like it to national funding. And then, of course, industry money, right? You need the industry money necessary to get these therapies over the finish line. And that finals, Arun was joking about glamorous, and you were like, glamorous what? It's a slog, right? And that slog, it looks very different from the philanthropic funding, high risk, high reward, proof of concept work, right? And it can't be denied that part of that difference is based on the fact that Novo Nordisk, in this case, sees great potential for human benefit, of course, but also has the coffers to get through these expensive trials and is able and willing to assume that risk, presumably because there's an expectation of major profit, right? I mean, I think that goes without saying, not cynical at all. While I understand the necessity for this virtual cycle underlying research, I mean, that's the way it's always gone. That's how you fund research development and ultimately translation of any drug or therapy, pretty much, when you got to do these large trials. But the cynic in me, of course, has doubts about companies assuming custody of these therapies. You just got to look at like the last 50 years in the drug industry, and you see this persistent theme of profits being put over patients. So, I mean, I get it. Again, I'm not a child. You got to do it. We got to get these therapies into people, and there's no other way. We need this big money industry. But do you think, and I want to look maybe on the bright side of this, do you think there is a similar risk with cell therapies being handed over into the custody of these profit companies that are run by boards? Or because these therapies are curative, does the curative nature of a therapy, is there possibly for like a different framework and outcome that you could see? I know it's hard to predict. I hate to put you on the spot here. You're just supposed to do the science, but you got to think about this stuff, right?
So there's many questions in one there. And just as a side, it's not stem cell therapy for Parkinson's, for example, is not really curative. The disease continue to progress. It's the symptoms that are stopped, and the function of the lost cells are replaced by the neurons. But absolutely, the whole field changed when there was commercial interest in cell-based therapies for Parkinson's disease. This is a field that I worked in for a long time. When I entered the field, it was a dead field. No one was interested, either academic funding agencies or not companies at all. The companies are absolutely needed, both for their money and expertise in doing this, developing global therapies, and you can't get away from that. I think when the danger comes in is if the academic scientists start looking for either what the companies want, but it doesn't even have to be like that. If the academic scientists are looking for proofs to support their current therapies rather than to scrutinize all data very carefully. And somehow it can be so then when there's a lot of commercial interest in the field, and I've seen this happen to other fields, not stem cell therapy, but other fields close to mine, is that it can switch the field from being kind of explorative and discover new mechanisms, new findings to prove that you're right. So if you set up experiments to prove that you're right, then you're in trouble, I think, or the field is in trouble. And this is not only due to the commercial interest or involvement in this, but the commercial involvement is a contributing factor to maybe scientists wanting to prove that they're right rather than to take a more holistic approach and be open for interpreting wider data and new avenues.
Yeah, it's a complicated situation, a lot of players involved in this area, but I mean, regardless, this is an amazing time to just be in this field and to be doing clinical trials in the pluripotent stem cell field with so many quote shots on goal, so many opportunities, so many different avenues being pursued. And they're being explored. These, pluripotent stem cells are being explored in clinical trials for all sorts of diseases. We're focusing, of course, on neurodegenerative diseases here on this particular show, but we've chatted with researchers and investigators in the diabetes field and cardiovascular disease, just to name a few high-profile examples from around the world. But while things are accelerating and it's kind of a golden age in stem cell derived clinical trials, this concept of using pluripotent stem cells for clinical applications, as you've alluded to, has been around since the beginning. Right. I mean, but in your opinion, why is this time, 2024, the best time ever for stem cell derived clinical trials? Is it mostly just because we've had this foundation of discoveries to build on and we've reached this point kind of organically? Or is it the non-science factors, the regulatory, financial side of things that have helped enable us to get to this point? Is it a combination? So what is it about this year, this day and age, this golden age, that has enabled us to get to this point?
I think we reached it organically. There's been a lot of, so following the field from the first human pluripotent stem cells, there's a lot, as we discussed earlier, about how do you culture the cells, how do you control their expansion. But then we needed to learn a lot about these stem cells, how they self-renew, how they differentiate, how you make them, what does it mean to make them in one way or another. The next step was to make specific cell types. And that just takes a long time, and the field hasn't been mature to go into clinical trials until in these years when it has. So I think it's several discoveries in the field that has taken us to this point. Now, I think it's a bit of a catch-up effect, because I think there's many projects now. They're at this pivotal stage where they are ready for clinical translation. And then the first projects for us are already in clinical trials. The work that we've done, not the least the interaction with the regulatory authorities, will pave the way for faster translation for coming projects. So there is a natural progression in the field, and one needs to let things take the time that it takes to develop the best therapies. With that said, you can't wait forever, because there's a patient in need of cells, there's diseases that don't halt while we test yet another factor to make even better cells. And so it's kind of, in Swedish, we say that we have to rush slowly, meaning that you always need to get to the endpoint the fastest way possible. Sometimes that means that you move fast, but sometimes that actually means that you move really slow. And I'm really happy that the clinical trials that have been initiated and approved today have had it's been an immense effort on focus on safety of these cells. But what the field do not want is quick cells going to trials too quickly and posing safety risks. That's going to derail the whole field. So I think that in the end, we are progressing to global therapies as fast as we possibly can, even though it might seem slow.
Well, slow and steady, right? That's what wins. And I think, people who are affected by this disease, it's never fast enough, right? But I think that we're in an age now where many people who are affected by these diseases may have hope and have cause to be much more optimistic about seeing a treatment in their lifetime. And I think that that's realistic. Whereas I think maybe 20 years ago, naively, there was a lot of promise that maybe didn't have a lot of foundation. Now the foundation's there. And while this is the greatest time, as Arun said, to be in the field, I don't think this is peak time. I think we're still on the ascent. And that's what's so exciting. And, no better place than Lund.
Yeah, and as you say, patients are desperate, and I fully understand this, but one also have to remember that no one is helped by unsafe or ineffective therapies. So there's no reason to move ahead until this has been proven prior to going to trial. So yeah, slow and steady.
Yeah, I mean, all the people who have been negatively affected, and they're substantial by these snake oil mesenchymal stem cell clinics, some of them to the enucleation of their eyes, would say that they could do without fast and messy. But yeah, back to Lund, very rich history there, founded in 1666, like 50 plus years ago. That's forever. And as you would expect from such deep roots, has accounted for countless advances across many fields, science and medicine, artificial kidney, ultrasound, also, more general science, the inkjet printer, which circled back into now printing cells, Bluetooth. The list goes on and on. Indeed, apropos to this conversation, dopamine, as you probably know, was discovered by pharmacologist Arvid Carlsson at Lund in 1957. Maybe it's that ownership of dopamine that inspired you and your colleagues there to take on this incredibly ambitious lift of the STEM-PD trial. But I'm guessing it's probably more to it than just that history there, Dr. Carlsson. So tell me, what is it? Tell us, what is it about the environment at Lund that makes it well-suited to fostering the work you guys are doing there? A little plug for your university, for the trainees out there.
Yeah, I don't know if you're so far off by saying they were started by Rick Carlson and at those early times, but there's been a long history of developing explorative therapies for Parkinson's disease and advanced therapies. And these are the first fetal tissue trials were conducted here in Lund by Anders Björklund and Ulle Lindvall, and they've really set up a trajectory of translating experimental findings to clinical trials. So we have the basic science in place. It's an excellent university with, we have the Lund Stem Cell Center is a great place to work. But there's many centers that are as good as the Lund Stem Cell Center. But what we have done is close to the clinic. We have a tradition and history of working translationally. We work together in clinical scientists, research nurses, experimental scientists, regulatory scientists. So we really have a drive, and it's promoted and rewarded to take findings to clinical trials. And you guys are young, but when I started, this was kind of people call it a dead end. Don't do translation, you'll ruin your career. And then working at a center where this was promoted really helped you in being convinced that actually, well, it may be a dead end. I don't care. This is what I'm going to do, but it doesn't have to be a dead end. So, and now the zones are changing, and translational science is what everyone wants to do, or that's very promoted. But it was 15, 20 years ago, it was seen as a dead end.
Yeah, I mean, times have definitely changed, but really it's that foundation that we're talking about, years and years of rigorous basic science done at places like London around the world to actually get us to this point. And so thank you so much for being here and sharing your journey with us. It's really great to see where the field is right now. But before we're going to let you go, we're going to ask you a couple of non-science questions, sort of broader questions for our trainees to help pick your brain a little bit. So first, if you're actually not a scientist, and we're very glad that you are a scientist, what would you be?
Well, When I was growing up, I wanted to be a travel agent because I grew up in a very small town in rural southern Sweden, and I wanted to see the world, and that was the best occupation I can think about to see the world. I don't think I would have been a travel agent, but I think I would have done something that let me be out in the world exploring cultures and kind of global affairs and things like that.
Yeah, and I mean, that's one of the best things about being a modern scientist is indeed you get to travel the world, right?
Yeah, I love it. It's I have collaborators all over the world. You get to that gives you access to the best science, but it also gives you access to the funnest way to work and observe. I mean, I love going to Japan and observe how they run the labs there, how they, it's a way to be part of the world somehow. And I think I would have found that role, but in a different occupation.
Yeah, I totally agree with you. And so the last question we have for you, what's the best piece of advice that you've ever been given, professional or not, that you'd like to share with our listeners?
Yeah, I mean, I've had the fortune of working with many good mentors, but maybe Anders Björklund here in Lund that I worked closely with for many years. He's a very balanced person, and he once said that you never make it or break it with one thing. And I think that's a good piece of advice for scientists, because often you feel like you make it or break it if you get this ERC grant or the NICEF grant, or if you get a Nature paper and everything is kind of focused. If I get this Nature paper, everything will be fine. If I don't get it, everything will ruin. But in the end, it's not like that. You have many chances to perform. You have many chances to fail. You have your success in your career actually depends on all these things cumulative. So there are no make it or break it events.
Well, you certainly made our day with this conversation, Malin, and we've enjoyed it. And it breaks my heart to have to end it, but it's been a joy, and I want to thank you again for sharing all your insights, and keep on going there. I mean, this STEM PD trial is really a shining light for all of us. That and the other few trials for the time being that are out there. I think we all look toward them really avidly. Can't wait to see the results, and are really, honestly, more than anything, grateful, as you started to say at the beginning of this conversation, that you're putting it out there, just showing that we're being transparent and providing a measure and a template for us all to follow. So thanks again for sharing with us, and thanks for doing your good work.
Yeah, and thanks for inviting me. It's been great fun.
All right, you guys, that brings us to the end of our show. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast, or via email at info@stemcellpodcast.com with feedback or to suggest guests. Until next time, thank you guys so much for listening.